So I will today talk about physical properties of DNA, physical and physicochemical properties of DNA. So DNA, physical as well as chemical structure of DNA. For this understanding DNA, you need to understand about what a nucleotide is, what a nucleoside is. You know that DNA is the genetic material in living organisms. So we need to actually go ahead with that. So I'm sharing my screen. I hope you people are able to look at this. Are you able to see this?
Yes, sir.
Okay.
Yes, sir.
So the term nucleic acid, let me make it a bit bigger. Was coined by the Swiss scientist Friedrich Mischer in 1869 because they were discovered inside the nucleus of the cell, and acid because phosphate was detected in the presence of phosphoric acid. So the name became nucleic acid, and it includes DNA and RNA. Okay. So now let us look at the structure of a DNA and RNA. This is a nucleotide structure. So what you have over here is first a base. You have a base. Then you have a sugar molecule. And you have a phosphate group attached. So this is known as nucleotide, a base. This is the base. This base can be a purine. So the examples of purine are adenine, guanine. This is the structure of adenine and this is the structure of guanine. Difference is that in adenine there is an NH2 group which is not present in guanine. Here it is. There is an hydroxyl O group over here. The NH2 is present over here. Otherwise, their structures are similar. And you have pyrimidines, the bases. They can be thymine, this is a single chain thymine, or cytosine. This is in the case of DNA. In RNA, instead of T, thymine, you have uracil. This is RNA specific. Clear so far?
Yes, sir.
Okay. So this is the structure of a nucleotide. Now let us look what a nucleotide, nucleoside, and what are the phosphates present. So you have a nucleotide. The nucleotide has got this single phosphate, this sugar. It can be ribose, deoxyribose. In the case of DNA, this O is not there. The 2 prime, this is the, sorry, I've made a slight mess of this region here. So in the case of DNA, this is carbon 1, 2, 3, 4, 5. So it's a ribose sugar. But when the 2 prime instead of OH, there is this only H present, then it is DNA, deoxyribose sugar. The 2 prime OH in RNA, that is ribose, and 2 prime H DNA, that becomes deoxyribose. So this is essentially the structure. So when you, what is a nucleotide? It has got a monophosphate, it has got the sugar and the nitrogen base. When you talk about nucleoside, you exclude only the sugar and the base is a nucleoside. Are you clear about this?
Yes, sir.
Okay. So nucleoside, you will call them as adenosine. Nucleoside, you will call them as adenosine. Or deoxyadenosine in the case of DNA. So, and the nucleotide will be deoxyadenylate. And the guanine, it will be, the nucleotide is deoxyguanylate, and the nucleoside will be deoxyguanosine. Similarly, the same thing will happen with the pyrimidions. You have to understand these nomenclature. Now, if you look at the structure of a single-stranded DNA, the 5, this phosphate, this is the 5 prime carbon here. This is the 3 prime carbon. And this is the first carbon. So the base, say adenine, is attached to this sugar, deoxyribose sugar. That is then attached to this phosphate. So this is known as the 5 prime end. So the 3 prime phosphate is attached to the 3 prime phosphate is attached to the 5 prime of the next nucleotide. Here, the phosphate, it is attached to the sugar, and that is attached to the base. So gradually you can see in DNA, one after the other, 3 prime to 5 prime attachment, you have a single strand of DNA. Same with RNA also. Instead of deoxyribose, you have ribose. Now, how does the base pairing take place? The interesting thing to note is that, so this is the 5 prime phosphate, carbon, 5 prime carbon, and that is attached to this base. So opposite of A with double hydrogen bond always is a T, and opposite of G with triple hydrogen bond is C, cytosine. And so if this is the 5 prime end and this is the 3 prime end, there is these hydrogen bonds formed between the parallel. And the next, when you're talking about double stranded, you have the 5 prime end over here and the 3 prime end over here. And one is in this direction, the other is in the reverse direction. So this is actually known as anti-parallel configuration. The two strands are in anti-parallel orientation and they are joined by hydrogen bonds. This is an A, it is joined with a T. You can see it here, purines and pyrimidines. Here's a G with a C with a G, and a G with a T with an A. Clear?
Yes, sir.
Yes, sir.
Now let us look at the Watson and Crick model. 5 prime end, 3 prime end, 3 prime end, 5 prime end, again 3 prime end. And you have hydrogen bonding over here. So this bonding, this is actually the hydrogen bonding. So they are in this orientation, a spiral orientation. Goes on like this. So if it starts like this, it goes on like this. This is the structure. One side is the 5 prime, 3 prime, and this side is the 3 prime and 5 prime. This is a helix. Helix configuration. So when you have this configuration, you have two types of grooves formed. This groove is known as the major groove, and this smaller groove is known as the minor groove. This is the minor groove. This is the major groove over here. So distance between from here to here, you have 10 bases, 10 base pairs. 10 bases are hydrogen bonded. And distance is 34 angstroms or 3.4 angstroms. So distance between one base and the other base here, this is 3.4 angstroms. 3.4 angstrom is 10 to the power minus 10 meter. It is so small. Clear? Yes, no?
Yes, sir.
Okay.
Yes, sir.
Now we will talk about forms of DNA. So Watson and Crick, that was Watson and Crick's model. So the DNA molecule which Watson and Crick described was in the B form. Now we know that DNA can exist in several other forms. A, B or C and Z. A, B and C is a right-handed helix. मतलब दाहिने से घूमता है. But Z form is a left-handed helix. It is not naturally found. It is found in vitro under high salt concentration. B form is the major form that is found in the cell. So, per turn, per 360-degree turn, how many bases are there? In the A form, in the B form, in one turn from the top to the bottom, from here to here, from here to here, 10 bases, 10 base pairs. That is in the B form. The A form has 11 bases. C form 9.3 bases. Z form, that is in the other way around, it is coiled. It has got 12 bases. Clear? समझे? Yes, no?
Yes, sir.
सुनाई दे रहा है?
Yes, sir.
Yes, sir.
1 second.
Yes, sir.
হ্যাঁ বলো কি বলছো? হ্যাঁ হ্যাঁ নিয়ে গেছি, ক্লাস নিচ্ছি এখন। Okay. So now we come to the next law, which is Chargaff's law. Arvind Chargaff, he found that DNA from any cell of an organism should have a 1:1 ratio of pyrimidine and purine bases. More specifically, amount of G should be equal to amount of C. That is G के opposite में C होगा. तो जितना गुआनिडीन रहेगा उतना ही साइटोसिन रहना चाहिए. Okay. And the same with the amount of A should be same. He found out chemically that amount of G is equal to amount of C in a cell. Amount of A is equal to amount of T in the cell. So this is Chargaff's rule. He found out in human, if there is a 30.9 amount, if the percentage of adenine is 30.9, the amount of T is also comparable, 29.4 in human. If the amount of guanine is 19.9, the cytosine opposite to it was 19.8, which is comparable. A by T and GC ratio should be 1. This is particularly this is the Chargaff's rule. So I will stop today's class over here. Otherwise, it will be a overdosage. Do you have any questions so far?
No, sir.
Okay then आज छुट्टी, ठीक है मेरे लिए, मेरा क्लास दूसरे टीचर लोग लेंगे तो अलग बात है. मैं आज तुम लोग को इस क्लास का छुट्टी दे देता हूँ, ठीक है? ओके.